celebration of, in honor of Gobind, who, as Tom said, really has been a very dear friend to me. I did my graduate work with Buzz Baldwin at Stanford University in the biochemistry department. For those of you who are from Wisconsin, Buzz's father was Ira Baldwin, who was a very world-renowned bacteriologist here at Wisconsin. When I was leaving Stanford to go to the Whitehead Institute at MIT to be a Whitehead Fellow, Buzz pulled me aside and said, you know, you really should get to know Gobind Khorana. I'm going to write him a letter and tell him that you're coming. And so when I arrived at MIT, I, with that introduction, was able to reach out to Gobind. And to my delight, he embraced me and invited me over to have lunch with him. And therein started a tradition by which Gobind and I would meet on a regular basis for lunch. And Gobind would come with his brown bag lunch, and he would go into the lab and grab a beaker and fill it up with some water. And we kept trying to offer him a coffee cup or something, and he'd say, no, no, that's fine. And he'd take his water in his beaker and bring it back. And over those years, those lunches provided me with a lot of support, a lot of advice, mentorship, and very importantly, inspiration. Gobind is just an incredibly inspirational person for me at a very formative stage in my career. And he used to lecture me to make sure that I stayed in the lab. He said, you know, don't get too tied up in these committees. You need to stay in the lab. And it was advice that I tried to take for as long as I could. We would talk about helices. We would talk about disulfide bonds. We'd talk about membranes. We'd talk about folding. We'd talk about bacteriorhodopsin and, of course, rhodopsin. I'd like to tell you a story today that started at MIT, and it has to do with viral membrane fusion. This is an electron micrograph at the top of an HIV particle as it's about to infect a cell, and on the right-hand side is an intermediate in that process. HIV is an enveloped virus. That is, it has a lipid bilayer that surrounds it, its genome. The cell, of course, is surrounded by a lipid bilayer. And in order for HIV to infect a cell, or for that matter, in order for an enveloped virus of any type to infect a cell, the two cell membranes need to fuse. The two membranes need to fuse. They need to become one. And that's a process that's mediated by specific proteins that interact in a specific way to cause fusion. Membrane fusion is, of course, important for many aspects of life. In neuroscience, the fusion of synaptic vesicles with the postsynaptic cell is critical for neurotransmission. And this is one of my favorite micrographs, which shows an intermediate in fertilization, in which the membrane that surrounds the egg has fused and become one with the membrane that surrounds the sperm in order for fertilization to occur. We got involved in membrane fusion when Chave Carr was a graduate student in my lab, and we were actually studying alpha helices and coiled coils, and had no this was a pretty random event that got us involved in membrane fusion. And I don't have time to tell you the story today about how that happened, but from that, and based on the work, the pioneering work of Don Wiley and his colleagues on influenza hemagglutinin, taken together with Chave Carr's work on coiled coils in the hemagglutinin protein, and subsequently extending that work to HIV through the work of many people, and summarized in this review article by David Chan. We came up with a model for how it is that HIV infects cells. Shown on the left-hand side is the viral membrane down at the bottom. And there's a protein called the envelope glycoprotein, which consists of two units. One is GP120, which is the piece of the protein that binds to the receptors on the target cells, T cells. And those receptors are the CD4 molecule and the co-receptors. Underneath the GP120 protein is a protein called GP41. And it is a protein that's anchored in the viral membrane. And it's also the protein that is known to insert into the target membrane. And the puzzle was, how does that insertion occur? And based on a lot of work, which I'm just not going to have, I don't have time to show you, we ended up concluding that an intermediate in this process was one in which the GP41 protein extended out in a spring-loaded manner such that it could take its hydrophobic so-called fusion peptide and insert that fusion peptide into the target membrane, such that the protein at this point was a membrane protein in two different membranes at the same time. Following that intermediate state, there was then a relatively slow reaction, minutes, in which these helices would collapse on each other to form a trimer of hairpins. This trimeric structure would form a 6-helix bundle and a trimer of antiparallel hairpins, which would bring the N-terminus of the protein close to the C-terminus of the protein, and thereby bring the two membranes close together. Membrane fusion then ensues in an ATP-independent manner. So somehow the energy that's achieved when this process occurs is able to overcome the activation barrier for causing membrane fusion. And one is left then with a single membrane and the post-fusion state. This intermediate here, the so-called pre-hairpin intermediate, is a transient intermediate, and it has an N-terminal helical region, and this is actually the 3-stranded coiled coil, as it's called, and a C-terminal helical region. And others had shown that peptides, synthetic peptides that corresponded to these C-peptide regions, actually could inhibit HIV fusion. And indeed, one of those peptides is currently a drug called Fusion, marketed by Roche, and it's used to inhibit HIV entry. And based on a lot of work that again, I don't have time to show you, we ended up concluding that the way in which these C-peptides inhibit this process is by binding to their N-peptide region in a dominant negative manner, thereby preventing the hairpins from forming because they were blocked by these C-peptides here. We went on to solve the crystal structure of this complex, and what we saw was that on the N-peptide coiled coil, there was a deep hydrophobic pocket that was highly conserved. And Debbie Eckert, together with her collaborators, was able to use a method that we called mirror image phage display to identify D-peptide, cyclic D-peptide inhibitors that actually bound to this hydrophobic pocket. This diagram shows you the representation of a co-crystal structure of the N-peptide coiled coil, shown here in blue, and the D-peptide, the cyclic D-peptide inhibitor. And so by X-ray crystallography, this inhibitor was shown to bind to the N-terminal coiled coil. And in cell culture, it was shown to inhibit HIV entry in a broad spectrum manner. And so that then led to the notion that you could inhibit this process by binding to that N-peptide coiled coil, either with the C-peptides or with these short D-peptides, as shown on this left-hand panel. That then led to the question whether or not, given that the endogenous N-peptide region is a target for drugs or drug candidates to inhibit this process, would this corresponding C-terminal endogenous region also be a target for inhibition? So could one, as shown with this box, bind to the C-peptide region and inhibit the intermediate in an analogous manner? So Michael Root and Michael Kay, when they were in the lab, designed an inhibitor that would bind to that C-peptide. And their design, I think, was simple and elegant. And that was to take the 6-helix bundle here and remove one of the helices and make a 5-helix construct, which would then bind tightly to a single C-peptide. And so they constructed a construct in which you had 5 of the 6 helices present, linked together with loops, and that left a C-peptide binding site. And when this molecule was added to HIV infecting cells, it inhibited HIV entry very potently. It was a nanomolar inhibitor with also broad spectrum activity. So taken together, this and a lot of other studies actually, we think, validate that this intermediate, the pre-hairpin intermediate, does exist as a transient intermediate in the fusion process, and that it is a validated target for inhibition by potentially therapeutic molecules, as shown here with the C-peptides, the D-peptides, or 5-helix, and another one which I haven't shown you. So the thing that this work all led to was a dream. And the dream was, could you actually target, could you use this target as a vaccine target? So instead of using peptides or small molecules to inhibit this process, could you immunize with something that looked like the pre-hairpin intermediate and thereby elicit antibodies? And those antibodies would then be able to go in and inhibit fusion the same way these peptides that I've been telling you about do. And what we had was the question of, could you raise antibodies, for example, to this region here, the N-peptide region? And we had things like 5-helix, which were well folded and exposed the N-peptide region. So could you use this as an immunogen to raise antibodies? Or we had other models where we had actually taken the 3 N-peptide helices and joined them with another linker which solubilized them, and could you immunize here and raise antibodies? And the idea, of course, is not to raise antibodies, but rather to immunize people and to be able to inject them with a synthetic peptide or with the 5-helix and thereby elicit an antibody response which would inhibit HIV infection. Well, that dream is now many, many years old. There were about 5 or 6 years of very hard work, both at MIT, in my lab, in several other labs around the world, and also at Merck, where we started a collaboration with the Merck vaccine group when I was still at MIT. And over 5 years of negative results, and really polyclonal sera after polyclonal sera, guinea pigs, rabbits, monkeys, nothing that showed anything that had positive results. And so that led to the notion that maybe this area here is too sterically hindered to allow an antibody to get in place. So maybe there just wasn't room for an IgG to get in place, because we were able to raise very good antibodies against these immunogens. They just didn't inhibit HIV infection. Well, before we threw the towel in for good, what we decided to do was to see whether or not we could select a monoclonal antibody as a proof of concept. So we went to a phage library, and we collaborated with a company called CAT, which makes very good phage display antibody libraries. And we used molecules such as these down here to select for monoclonal antibodies that bound to the N-peptide region. And then we screened those monoclonal antibodies to see whether or not they would inhibit. And we were able to identify monoclonal antibodies which did inhibit HIV entry. And so here, in a paper that was published in 2005, we show the results of an antibody called D5, which was discovered in this phage display methodology, that as a full IgG is able to inhibit different strains of HIV from entering cells, but weakly. These are hundreds of nanomolars for the IC50. So the activity was broad, but weak. Nonetheless, what this showed was that it was possible to raise antibodies against this region, and that those antibodies were, in theory, able to inhibit HIV entry. The group went on to determine the crystal structure of this antibody bound to 5-helix. And what they found was that the antibody indeed bound to 5 helix, and in fact it bound to that hydrophobic pocket region that I showed you. And then they went on, this is the crystal structure of the antibody, shown bound to the region of the 3-stranded coiled coil. And importantly, when you look and model the space that's available to this antibody, you see that even with just the coiled coil alone, there's enough room for an IgG to get in there. Never mind the additional parts of the protein which are missing from this model. So this monoclonal antibody and the crystal structure, which showed that in fact it did exactly what we thought it was doing in terms of where it bound, reinvigorated the effort and reinvigorated the project. It's sort of, I think it's one of those things in science when you've worked for years and years and you don't get anything, and then you're about to throw in the towel, you do that one last experiment, and then you're reinvigorated. The project has been reinvigorated, and the work is ongoing right now. And the sort of approach that we're taking is to stabilize the trimers using disulfide bonds, to shield the part of the peptide which is used to solubilize things from the immune response by putting short polyethylene glycol linkers on there, and to extend the length of the peptide that corresponds, that comes from the HIV portion. And at this point in time, what I can say is that we actually have now polyclonal responses which are neutralizing HIV entry. The responses are still weak, but I think invigorated by the fact that it's possible, one actually looks harder and tries harder to find the polyclonal responses, and some of these immunogens now are giving us responses. Still a long way to go from anything that would be able to, that would satisfy the criteria that we would want to test in humans. But we're now going into non-human primates and trying to test these candidates to see whether or not we can elicit antibodies. And we certainly hope that as we publish this work that will stimulate other groups to reinvigorate their own efforts to try and come up with antibodies against this part of the protein. It's important to do this. This shows the United Nations summary of the situation with regard to HIV in 2007. The 2008 results are going to be published shortly. In 2007, 1 in 200 people living on Earth are infected with HIV. There are roughly 6.5 billion people on Earth. 33 million are infected, so that's 1 in 200. Merck had another vaccine in clinical trials which used a completely different concept, and that was to elicit a cytotoxic T lymphocyte response. And so using a replication defective adenovirus that contained pieces of the HIV genome, namely GAG, POL, and NEF, we were able to elicit cytotoxic T lymphocyte responses in primates, in non-human primates, that were able to keep the viral infection under control. It didn't prevent the viral infection, but it kept it under control. But we went into 2 large phase 2 studies in humans. The vaccine failed miserably. And so that effort, together with many other efforts aimed at eliciting a cytotoxic T lymphocyte response using that sort of an approach, are now on hold, and scientists are trying to figure out how to get a cytotoxic T lymphocyte response back into clinical trials. In the meantime, it's our hope that something along the lines of what I've presented to you today, namely going after an antibody approach and trying to target a conserved region which is not normally exposed and so therefore would not be subject to the large variation that other parts of the protein are, might be a potentially promising avenue. And so with that, I'd just like to end by saying that the work that I've described is actually the work of a lot of people. These are just the major contributors, both at the Whitehead Institute at MIT and at Merck. And Gobind, it's great to see you again. It's great to have this symposium in your honor, and thank you very much to the organizers for the invitation.